Welcome to Brain Talk, everyone. Now let's talk brains. Dr. Ahmed Abdelfattah is here today to talk to us about exciting new technologies to study brain cells.
So a little bit about myself. I'm a biochemist by training. I'm really fascinated by how proteins work, but I'm also fascinated to apply my knowledge in biochemistry to the brain, and that's what I'm going to be sort of focusing on today. So, my talk is about new ways researchers are developing to study how the brain works. And a human brain, for example, has about 100 billion neurons, and each neuron has over 10,000 connections. And that goes back to what Carla and Victoria were saying previously. Like, we have more connections in our brain than connections in wires are making in this on Earth, or stars in the Milky Way, and so on and so forth. So it's really important to try to sort of segment and dissect how these things are working in our brain. We know that brain cells communicate using electrical and chemical signals, and measuring those signals is crucial to understanding brain function. Every thought, action, memory is encoded by changes in those electrical and chemical signals in our brain. So if I move my arm like this, my brain changed there was some electrical signal to my arm muscle from my brain for me to move my arm, and so on and so forth. One method that many of us might have heard about in recording how the function in the brain is our functional MRIs, where doctors measure blood flow to certain areas of the brain, and because blood flow is correlated with brain cell activity, then you can deduce which area of the brain is active. This is extremely useful in many instances, and it gives you a general idea of the activity of the brain, but it's also very coarse and grainy. You can think of it as, every few million neurons or every few million brain cells are show up to you on a screen as one pixel. So you don't really see what's happening within those few 100 million neurons in your brain. There is no really good way using fMRI, for example, to tell you which brain cell or which brain circuit is involved to move your arm, for example. So to improve this resolution, scientists have used electrodes to record from individual cells. This is extremely useful technique and has revolutionized how we think about the brain, and allowed us to monitor brain activity at extremely fast speeds, but it's invasive, right? You have to impale a cell with an electrode. It's an extremely laborious experiment to do, and you can record from 1, 2, or 3 cells at a time, so it doesn't really give you a global view of how the brain is working. So to try to see individual cells, how they're acting, what their chemical and electrical signals look like, without being too invasive, we can turn to fluorescence microscopes. So the trick is to get the cells to change their fluorescence properties when something happens. So, if you look at this wire, right, you can't really see the electricity flowing through it. So you have to have some sort of contrast agent. You have to make a contrast agent to label the cells with these molecules to change their fluorescence properties when the brain cell is active. And I want to quickly show you a movie here of one of the examples we've made recently towards this, where we basically label, this is a neuron in a mouse brain, and we label its membrane with a molecule that changes its brightness when that changes its fluorescence intensity when the neuron fires an action potential, or when there's an electrical signal in the cell. So on the bottom here, you can see the electrode signal if you actually put an electrode to measure its activity, but you can also see that the fluorescence, or the light that's coming out of it, is changing over time, and it can report the individual changes in membrane potential in this cell. And in the same vein of this, we're making also, we and others are making other tools to record other types of chemicals. For example, like Carla has mentioned, we're making some tools for opioids, for example, to see where opioids are acting, which circuits in the brain it's acting on, and so on and so forth. So that's a sort of an overview of what we're interested in and where the field is going, and I'm happy to take more questions.
Thank you, Ahmed. So I'm kind of curious how you got into this field in the first place before we get into the deep science.
So I'm a biochemist by training. I'm really interested in working with proteins, and originally I wanted to apply my knowledge actually. So I got a Ph.D. in chemistry from Alberta, just the state next to where Carla studied in Canada.
Oh, wow.
And I was really interested about proteins. I didn't know anything about the brain. I love chemistry, and I liked how proteins use chemistry to do many, proteins are the workhorses of life. Where every function we do, our cells do, are done by proteins, right? I originally was working in a protein-focused lab, and then I learned that I can make protein tools and put them in cells in a culture and see, repurpose the proteins in nature and make them useful in the cell to show me of the function of the cell. So the first protein I worked on actually changed calcium concentration into a fluorescent signal. So when the calcium concentration changed in a cell, this was a cancer cell on a cover slip, we can see the calcium concentration changing over time with that protein because it's changing the calcium concentration into a light output. And I was like, that's really cool. And then I started collaborating with some neuroscientists to see, and I learned more about the brain, and I was like, that's a really cool organ to study. And I'm trying to apply my, my protein engineering toolkit to study the brain.
Yeah, that sounds very familiar. I always tell my students that all roads lead to the brain, because no matter what your PhD is in, or no matter what you actually studied in college, there are pathways to study the brain. I actually in college, my bachelor was in fine arts, so there you go. But yeah, I mean, whether you started chemistry, or you studied physics, or you studied biology, or you studied computer modeling or mathematics, right? All of these paths can eventually lead to investigating the most complex organ on the planet. I'd also like to get sort of like even farther back in your background, right? Because one of the purposes of Brain Week is to really inspire the next generation of scientists, right? So, growing up as a kid, what was your access to science? Were you that kid that always knew you wanted to do science? Who were your mentors? Like what happened along the way?
What happened
Asking you to get
along the way?
deep here.
So no, I was not the kid that always wanted to be a scientist. So I grew up in Egypt, where research is really limited. In fact, I don't think there's any brain research there because it's too expensive to do any of those experiments. Like only research in Egypt, for example, are engineers because they can do everything on a computer, right? So I didn't have any access to sort of research beyond me learning about, some basic chemistry in class and so on and so forth that I really enjoyed. I really liked chemistry when I was a kid, but I also liked sports. I was playing sports more than I was liking chemistry. But then in college, actually, a couple of my professors, and I still actually am in contact with them now, really made me like how chemistry can be applied to biology. And I remember we took this pharmacology class, and it was really fascinating to me how so my bachelor's is in pharmacy, so I have a pharmacy degree as an undergrad. And how those small molecules, like we were building small molecules in an organic chemistry lab, how these can change the function of cells theoretically in a book, right? I never did it in the lab. So I was like, that would be cool to do it in a lab at some point. And that's how it got started.
Right. And now you are at the cutting edge making tools that
presumably are going to change the nature of how we study the brain. Will you tell us a little bit more about the tools that you specifically are working on? And I think a lot of people are interested in knowing about, what are the economic implications of this, right? So biomedical advance is part of the Rhode Island economy, if not the national economy, right? So how do you see your tools fitting into that sort of economic environment, which is one of the areas of growth in our state?
Can I tag onto that? Could you also talk about, because I think your tools are amazing, for to understand on a very, very microscopic level what neurons are involved with certain behaviors or things like that. What are the challenges? I mean, we've been at it for years now. What are the challenges that you've had to overcome while developing those tools too?
Yeah, so okay, so to start, maybe I'll talk a little bit about the types of tools we are actively making in the lab now. So we're focused on three main sort of themes in the lab. One is building a better toolkit for voltage imaging, so recording electrical activity of the brain. And this comes with a lot of hurdles. And one of the hurdles is that to record electrical activity, the place in the cell where the electricity is being conducted is the membrane. So the membrane is actually an insulator, and really close to the cell on the outer and inner leaflet of the membrane, this is where the most of the electricity is being conducted. And you have to have your probe there and not in the rest of the cell. So imagine that you're already very limited in where you're recording from in a cell. So imagine you have a soccer ball and you only can record from the surface of it. You can't record from anywhere else, right? So to record from there, you need your sensor to be really bright, really giving you a lot of photons so that you can see on a fluorescence microscope. You need it to be, so to do that, you shine a lot of light, but then you can, light can, at very high quantities, can actually damage your tissue. So you want it to be bright enough such that it doesn't need a lot of light. So that's one thing that we're working on actively in the lab. The other thing is,
can I insert something there just for the purpose of the audience? Because I think, and you kind of talked about this, but I think it's so important for people to understand how important electricity really is in the brain. We know a lot about pharmacology, right? That area of drugs, and that if we, alter the chemical imbalances in our brain, then that's what's going to cure diseases and that's what's going to treat us. But electricity really is the language of the brain. I always tell my students that the chemicals are just the messengers in between the neurons. And so I just like to reiterate to the audience how important this work is that Ahmed is doing, because electricity is the way that we are going to figure out what is wrong with cells in disease and how do we treat those cells in disease. So sorry, go on.
I like to think of it. So I like to think of it as like, if you record the electricity in the brain, you're watching TV in black and white. You will know what's happening. But if you want to put the flavor, like if you want to watch TV in color, you start watching the chemicals, which chemicals are involved, right? So,
I feel kind of salty right now because I like the neurotransmitters.
No, but yeah, it's important. Like if you want to put color and understand really, what is responsible for this electricity moving, being produced and so on and so forth, you need to know some sort of the chemical neurotransmission in the brain. And that's sort of the second theme that we're working on in the lab. So we're actually focusing on neuropeptides, including opioids and other neuropeptides like substance P, which is involved in nociception and pain and so on and so forth, and trying to make probes for those to, again, change. So when there's no way right now for researchers to know which individual cells are releasing those neuropeptides and which cells are being acted upon with these neuropeptides at a cellular level. We can say with microdialysis that this general region of the brain you stick an electrode, you stick a probe, and then you can analyze the chemicals in this region of the brain. And you can say there's high concentrations of this, high concentration of that, but you can't really say at a cellular level which cells are releasing or being acted upon with this neuropeptide.
Right.
And,
which is important to understand when we're trying to understand the human brain, because these
exactly.
neuropeptides are everywhere and
Exactly.
they are used for different behaviors in different areas of your brain and in different neurons.
Exactly, and actually, so new data is showing that, so there's lots of like sequencing data coming out now of many different neuroscience labs, and they have shown that at a single cell sequencing, so if you get a neuron from your brain and sequence it, every cell in our cortex or in a mouse's cortex expresses a neuropeptide. But people don't really like, so opioids, for example, are being expressed in the motor cortex, but people don't really know because it's not being released at a high enough concentration to image it with a microdialysis probe. But when we have those sensors to see at a cellular level, maybe we can be able to see those probes those neuropeptides at a cellular level.
Yeah.
So
no, and I always talk to my students about this, because I say, when we think about treating mental illness, for instance, right, treating schizophrenia, we think about medication and drugs, right? And so antipsychotics, for instance, part of what they do is they modify the dopaminergic system, right? And so we think, okay, dopamine, that's going to take away your hallucinations. But the thing is that, like you're talking about with this neuropeptide, dopamine is everywhere in the brain, and it's used for many other reasons. So for instance, your ability to move, to initiate movements in a coordinated manner depends on dopamine, right? And so when you take a drug to solve one problem, you are causing other problems because you're disrupting the dopaminergic pathways in other places, right? And so somebody once told me, the analogy to using drugs, pharmacological agents is, if you had to put oil in your car, you would open the bonnet and kind of like just dump oil everywhere, rather than opening the oil chamber and putting it specifically in there, right? And so the tools that you're talking about, you're basically talking about finding ways to tailor the measurements and the whatever changes that you want to make to a specific cell, and not just like every single cell that expresses neuropeptide or dopamine.
Yeah, the hope is to be able to understand which cells are involved and then only target those cells in the future, so that you don't have all of those side effects of it's like hitting a hammer basically, instead of like individually knowing where you really need the action of your drug to be. So that's true.
I just finished reading a book called Peak Mind by Amishi Jha, who talks about mindfulness and the way meditation and mindfulness can actually change the chemistry of the brain. And I'm wondering if you've done any of that work or are aware of any work being done in that area.
I haven't done any work in that area. I mean, sort of our lab is more of a tool development lab rather than that, but I'm sure people are doing that here in Rhode Island and elsewhere.
Yeah. How close are we to applying these technologies to humans? That's always what audiences sort of want to hear about, right? Because we're the nerdy types being like, oh, let's do this in fruit flies and mice, but to normal humans, they're just like, all right, so when can you take my brain and modulate a specific cell in my brain?
So we're not close. That's not going to happen, I think, in my lifetime. But I want to say that it's really important to try to understand simpler brains, like a mouse's brain or a fly's brain, so that we can extrapolate what we learned there to a human brain. We don't necessarily need to record from a human brain to understand how the neural circuits in the human brain work. We know the analogous circuits. We can draw correlations between animal models like mice, like flies, to a human brain. And the hope there is to find, drug molecules in animal models that can be tested in humans without needing to record from humans right away. So that's how it's, I think, will work, at least in my lifetime. But
Some people who are more idealist. But yeah, I think you're right. I think
we have some questions from the audience. How do you think the field will change in 10 years or 50 years?
I believe that optogenetics, or the use of light to record and manipulate cells in the brain and other areas of the body, really, will take over from once we get it right, once we get the right tools, once we are able to find tools that use the right amounts of light so that we don't damage cells and so on and so forth. We don't really need to use any, there's no real need to use any mechanical interventions to an animal. So we can imagine an animal is going about their behavior in a whatever setting they are doing, and then we can use light to record how their brain is functioning in its natural environment. So in 50 years, that's what I hope. Like, you can have a mouse in a forest and you're recording its brain activity in a natural setting. That's sort of the hope. That's my dream, at least, and we'll see how it pans out.
Could you speak a little bit more about optogenetics? Because I think that's it's a tool that is very exciting for scientists, and we've known about it for the last decade or so. And that's one of those technologies that when they make their way into TED Talks, people are like, this would be amazing to do in humans. And it's probably the closest technology that you could realistically, maybe some way down the line, hopefully, perhaps. So
optogenetics is really like opto, which is light. Genetics is using genetic targetability for expressing whatever you want to express. So it's light, using light to control or record from genetically targeted cells. So light is fine. I mean, you can envision using light on a human brain, but the genetics part, introducing a gene in the brain of a human, is harder. So how it's done in animals is that we can use, we do this for a living in our lab. We find proteins which are encoded by a piece of DNA that we put in a mouse brain, for example, or a fly brain or a zebrafish brain. And this DNA, the genetic part of the optogenetics, will encode this protein and express, let's say, on the surface of the neuron, like I showed you in the movie, and it will change its brightness when the cell is active, or so we can record the electricity from the cell using light. So that's optogenetics. We can also have that thing on the membrane not be bright, but actually when you shine light on it, it can activate the cell. It can open a channel and sodium rushes in, and then it can activate the neurons that you're shining light on. So you can have actuators or sensors, and the combination of them with light is what optogenetics is. And you can do this pretty reliably now in animals. But the toolkit, I mean, to multiplex different sensors and to find the right different wavelengths for different sensors and so on and so forth, is tricky and requires some work. And that's what we're doing in the lab.
Right. So, I mean, basically what you're doing with optogenetics is you're taking a little laser beam and you're using a little laser beam to turn on or off a cell, right? But in order for that cell to be able to turn on or off, you have to have genetically modified that cell in order for it to be responsive to that laser beam, which is something that we do in science, in animal models pretty reliably. But if and when we ever get that in humans is the big question.
Yeah, I think.
Sorry.
Piggyback on that real quick is that that's while we do this kind of work, it's pretty exciting work. There's also the ethical
exactly
thing, in doing them in
Great
humans.
minds.
And so I know that's a big challenge because you have to, introduce something foreign into the human body and you target in the right position. I know that's some of the things that's delayed using some of this technology in humans. But some of the ones that are out there already, is there ways that we can already use some of the ones that people already use for like functional MRI and make those tools a little better? that you have more precise imaging of certain regions and things like that? Is that something that you're also working along in parallel to what you're doing right now?
So for functional MRI, it's a totally different ballgame. The resolution is very different, right? So you have a few 100 million neurons in one pixel.
Yeah.
So it's you really need light, and you need the genetic tractability, because if you only have light and you don't have a gene being expressed in a specific subpopulation of neurons, you can think of my brain as an orange, okay? and
I'm never going to unsee that.
Think of my brain as an orange, okay? And you put a fluorescent dye on the orange, everything like this whole orange will be purple, let's say, okay? You don't see individual parts of the orange changing in color because everything is purple, right? It's very hard to tell which cell is working.
Yeah.
But let's say you have pockets in the orange where you can put the purple dye, right? You can see the pockets of the orange, right? You can see those pockets in different color versus the rest of your sphere, right? So if you don't have the genetic part of it, it's very hard to implement. So the ethical part in humans is how to get an extra gene being expressed in a subpopulation of cells, and that's the tricky part.
All right, so we are at 1 minute, but I did want to ask one more question that I think is very important and is on the minds of people when they think about new tools for the brain. And Timmy sort of started to talk about it, but ethics, right? So when you are developing tools, you are, at an Ivy League institution, Brown University, and the tools that you are creating now, maybe not in your lifetime, but at some point will be used for other purposes that are not the ones you intended. So, does your lab think about ethics? Does the institution think about ethics, and sort of what conversations are being had in general in the field about ethics?
Yeah, I mean, the institution and our lab definitely think about that, and we make sure that we develop tools that are, we make sure when we apply them in animal models, for example, and so on and so forth, we apply them for a reason and not just applying them to test things that might not pan out. So we have a reason to test in animals and so on and so forth. Tools being used outside their intended use case is true for any field, really. So there's no way for me or even Brown to stop that, but I think it's very hard to apply the sort of tools we're making now into humans now because of ethics reasons. No doctor will allow that to happen, at least hopefully in my lifetime. And also those proteins are, it's if you express the proteins we're making, the tools we make are proteins. So if you express any other protein in the cell, it will be the same problem you face. So it doesn't have to be, our tools don't have a specific thing that will change or modify the cell versus another protein, for example. I hope that kind of answers your question. But
yeah, I think overall, you're saying that there is already regulation in place in terms of the ethics of what we can do in humans.
Absolutely.
And so
yeah,
and that will continue to develop. And I know that there are organizations that are very concerned with this and talking about it, and those discussions are being had. Thank you so much, Ahmed, for being with us today, and happy Brain Week.